There are 5 basic reagents or ingredients used in PCR: template DNA, PCR primers, nucleotides, PCR buffer, and Taq polymerase. The starting DNA is known as template DNA. Template DNA is the DNA that is amplified during a PCR reaction. Only a small amount of DNA is required for analysis. Just like cellular DNA replication, PCR requires a primer to get started. A PCR primer is a short piece of DNA that identifies the region of DNA that will be amplified during PCR. The PCR primer can bind to a specific complementary sequence of DNA. Primers are typically used in pairs, and the DNA between the 2 primers is amplified during a PCR reaction. DNA nucleotides are formed by pairs of adenine and thymine or guanine and cytosine, bound together by hydrogen bonds. The Taq polymerase uses these free-floating nucleotides to assemble new copies of the DNA segment being amplified. The PCR reaction is carried out in a buffer that provides a suitable chemical environment for activity of DNA polymerase. The buffer pH is usually between 8 and 9.5. For Taq DNA polymerase, a common component in the buffer is potassium ions from potassium chloride, which promotes primer annealing. Once the reagents are placed in the thermocycler, 3 phases occur to amplify DNA. During the denaturing phase, the double-stranded template DNA is heated to separate it into 2 single strands. During the annealing phase, the temperature is lowered to enable the DNA primers to attach to the template DNA. During the extension phase, the temperature is raised and the new strand of DNA is made by the Taq polymerase enzyme by putting nucleotides onto the strands after the primers. During each cycle, the amount of DNA doubles. Therefore, if you were to start out with 1 strand of template DNA, you would end up with 4 strands of DNA at the end of the 3 cycles. Essentially, the template DNA splits up, and then each time it is amplified.
There are 2 types of DNA replication. There's in vivo and in vitro. DNA replication is carried out in vivo at a body temperature of 37 degrees Celsius with the help of several proteins and mechanisms. The helicase unwinds the DNA. The DNA's single-strand binding proteins stabilize these unwound strands, etc. And in vitro, the PCR reaction utilizes temperature cycles to cause denaturation and annealing of the DNA strands. In vivo and in vitro replication differs by polymers. There are many DNA polymerases in eukaryotes. In PCR, thermostable DNA polymerases derived from bacteria or archaea are used, i.e. Taq polymers. In vivo, in vitro DNA replication differs by length of DNA. The whole genomic DNA is routinely replicated in the body. In the PCR reaction, the polymerase used has a much shorter half-life and is only efficient for smaller fragments of DNA. In vivo and in vitro, DNA replication differs by features of polymers used. High fidelity, speed, proofreading, and repair are desirable features required of DNA replication. PCR reactions use simpler polymerases that are not as feature-rich. Gel electrophoresis is a method for separation analysis of DNA fragments based on their size and their charge. This is the only way to analyze the products of gel electrophoresis. Following PCR, you can take the amplified DNA from each of the samples and place them in an agarose gel, or this rectangle, or square. The electrical current will sort the DNA fragments from different samples by size, small and large. This allows you to quantitatively compare DNA fragments collected from PCR.